Fertilization to the bilaminar disc. In this course, I begin to explain embryological development of the spinal cord and vertebra with the bilaminar stage of a fertilized egg. In your folder, there are 3 videos that cover the stages of development in a fertilized egg from zygote through trilaminar disc. If you do not have any background in the stages of human embryology, I suggest you watch those videos before studying the material in this module. The bilaminar disc is made up of 2 layers, the epiblast layer and the hypoblast layer. The hypoblast layer gets its name because it's under and therefore hypo to the epiblast layer. From these 2 layers, the cell will transform into 3 layers, forming the 3 primary germ layers of the embryo. The primitive streak appears as a localized linear thickening on the dorsal surface or outer surface of the epiblast layer of the bilaminar disc. The primitive streak first appears at the caudal end of the embryonic disc. How do we know it's the caudal end? Because as we watch the stages of development, we see how the location of the primitive streak establishes body part orientation. Body axis. The primitive streak defines the major body axes of the embryo, such as the cranial end from the caudal end and the left and right sides of the embryo. Now let's talk a little bit about terminology. When referring to the cranial end of an embryo, I can also use the terms cephalic end and or rostral end. All 3 terms, cranial, cephalic, and rostral, refer to the head region of the embryo. So please do not let my interchanging of these terms confuse you. The primitive streak elongates towards the cephalic, also known as cranial, also known as rostral end of the embryo. At this end, the primitive streak forms a circular depression called the primitive node. Some textbooks also refer to this circular depression as Henson's node. Invagination. The elongation of the primitive streak towards the cephalic end of the embryo is caused by epiblast cells migrating through or falling through the primitive streak into the median plane or median space of the bilaminar disc. This migration of epiblast cells is called invagination. Invagination is the event or process that initiates the formation of the 3 primary germ layers. To reiterate, the appearance of the primitive streak signals the beginning of gastrulation. The elongation of the primitive streak indicates the invagination process has begun. Formation of endoderm. The first wave of epiblast cells to invaginate through or migrate through or fall through the primitive streak will head straight for the hypoblast layer of the bilaminar disc. The epiblast cells will wiggle their way in and around the hypoblast cells, eventually displacing the hypoblast layer. Once the epiblast cells have completely displaced the hypoblast layer, it is now called endoderm, one of the primary germ layers. One concept you need to wrap your head around is that during development, cells are constantly differentiating into new types of cells with different properties. So as in this case, the merging of the epiblast cells with hypoblast cells created a whole new cell or layer of tissue called endoderm. Formation of mesoderm. After the formation of endoderm, epiblast cells are continuing to migrate through the primitive streak. These additional epiblast cells invaginating through the primitive streak will form the mesoderm layer. Formation of ectoderm. Once the mesoderm layer is formed, invagination of epiblast cells ends. The primitive streak stops elongating towards the rostral end of the embryo, and the epiblast layers of cells differentiate into ectoderm. Now all three primary germ layers have been established, and the embryo is now called a trilaminar disc. So what you have witnessed here is that epiblast cells give rise to all three primary germ layers of the embryo. So to reiterate, the formation of the three primary germ layers is the main goal of gastrulation and the most significant event in the third week of development. From these three germ layers, all body tissues and organs and cells develop. We now are going to look at what these individual layers give rise to as the embryo develops. The fate of germ layers. Ectoderm gives rise to the epidermis of skin, hair, and nails, the brain and spinal cord and neurocrest cells. In this course, we will focus on the development of the brain and spinal cord and neurocrest cells that arise from ectoderm. Endoderm gives rise to the epithelial lining of digestive, respiratory, vascular, and urogenital systems and associated glands. Mesoderm gives rise to all other structures of the body not formed by ectoderm and or endoderm. So when in doubt, pick mesoderm. The two main structures we will discuss in this course that arise from mesoderm are the notochord and soma. Here is a chart of structures that develop from the three primary germ layers. I thought it was a good example to see what different tissues arise from the three primary germ layers. A sacral coccygeal tetraoma is a tumor that results from a persistent primitive streak. You see, under normal development, the primitive streak diminishes to become an insignificant structure in the sacral coccygeal region. If a remnant persists, a large tumor forms called the sacral coccygeal tetraoma. These tumors typically affect females, 75%, more than males. I could not find out why there's a gender preference when researching this condition. Typically, this type of tumor will be surgically removed soon after birth. Notochord formation. As mesoderm layer is being formed, some mesodermal cells begin to aggregate immediately beneath the primitive streak. This aggregation of mesoderm cells form the notochord. Axial support. The notochord is the first primordial axial support of the embryo. Do not confuse the term axial support with body axis. Remember, the primitive streak establishes the body axes, such as the head, tail, left, and right. Axial support is different. Think of the two regions of the skeleton, the axial skeleton and the appendicular skeleton. The notochord is setting the framework for the axial skeleton, and around this structure, primordial vertebral bodies will form. The nucleus pulposus of the intervertebral disc, and we'll cover this more extensively when we cover development of the vertebra.